So today, it's my pleasure to introduce Dr. Jaime Modiano. Jaime is the Perelman Professor of Oncology and Comparative Medicine. He's the director of the Animal Cancer Care and Research Program. Jaime completed both his veterinary medicine degree and his PhD in immunology at the University of Pennsylvania. He then was a resident at Colorado State University, and he postdoc at National Jewish Hospital in Denver. And he tells me that we met in the 80s. I think I was a graduate student. Jaime was a postdoc or resident, right?
No, I was a grad student.
You were a grad student too.
It's a baby grad student.
So I probably had big hair. It was the 80s. And we met on our first meeting was on a cycling trip at a meeting at a Gordon conference. And we kept running into each other. We were both from Colorado. So I was delighted when he joined the faculty here. He's been a fantastic faculty here, as many of you know. He's a colleague to many and an all-around great guy, even though he's from Mexico City. We're not going to put him behind the wall or make him pay for it. We were discussing that before the seminar. So Jaime studies the basic mechanisms of how and why cancer and autoimmunity happen. And so how does that happen together? And he uses companion animals as well as human model systems to do this. And so he's a master at comparative biology as well. And so I'll let you go with that, I guess. Thanks. You can tell horrible 80s stories about him.
Oh, yeah, absolutely. Yeah, so Carol called me this morning, and I remember when I was a resident, and I had vet students at Colorado State, and Patrice and Michaela will probably relate to this. They would call in at 6:30 in the morning, hanging over upside the bed and saying, "Oh, I'm so sick," and you realize that they were so sick because of the bender they had last night. And I think that's exactly what Carol sounded like this morning. It's like, "Oh, the snow is terrible." Yeah, but anyway, I'm glad Carol made it to introduce me. And yes, we did meet in the 80s, and she was as hot then as she is now, if I can say that. I'm from Mexico, right? So I can say these things because I'm gonna get deported anyway. So I've got 10 more days, and then see you guys. All right. So actually, what I'm going to tell you about is an interesting a decade on story, and it's the product of a great collaboration that started about a decade ago with Aaron, who's actually sitting here, and when I came and I talked to him the first time about the stuff that I was going to do, it took him about half an hour to stop laughing. And then he actually started looking at the data, and I think he's a total convert now. So he thinks that this comparative stuff is pretty cool. And he has been one of the most rigorous people that I've ever met. And I think he really has been transformational in the science. The other person who is not here yet, and I hope she makes it because when you ask questions, I'm going to need her to answer, who's actually done a lot of the work that I'm going to tell you about, is Malka Scott, who's been in the lab for about 8 years. And in addition to being the lab manager, she has done some terrific work and really has been the driver for a lot of this stuff that I'm going to tell you. And the other two people that I think are important to recognize up front are Subri, who again has been a friend and a colleague and a vital part of this for the better part of the decade. And Hiro Tomiyasu, who was a postdoc in my lab for quite a short time, actually, about a year and a half before he got recruited back to go be an assistant professor at the University of Tokyo. But in the short time he was here, he actually made some major contributions. So today I'm going to tell you about, I'm trying to make sure that if I point to one screen, everybody can see, because I hate the little gizmo pointers. So I might go back and forth so that we're equally compromised. So I'm going to talk about how we've developed a very robust prognostic test for osteosarcoma. Actually, we've developed several, but I'm really going to focus on the latest one and how we got there. But most of what I'm going to tell you about today really is based on my roots as a pathologist, so trying to improve our ability to diagnose and prognosticate disease. And one of the frustrating things has been the obstacles that we've encountered to make this a widely adopted test. And really how this brings us back to reality from research that we think is really important to the practical nature of making things that can go into the real world. And how these obstacles really spurred the development of something that will become more user-friendly to do biomarker discovery. And hopefully that biomarker discovery can then be more easily translated. So the take-home message that I want you to have by the end of the talk is that we have developed a new model for biomarker discovery that really takes advantage of this comparative approach. And it's not necessarily based on whether we have companion animals or not. The idea is that we can actually use this comparative multi-species approach to discover some pretty cool things. The process that we've developed utilizes blood samples as opposed to biopsy samples. And for those of you who are in the clinic, you know that biopsies are challenging. I mean, we all think that it's easy, but it is quite challenging to actually go and do these invasive procedures on patients. There's all sorts of issues of quality control, et cetera, whereas blood is actually quite rarely obtained, much more easier to handle. And that this model can be used to uncover biomarkers for early detection, to define biological behavior, and potentially to monitor response to therapy. Okay, so there's some disclosures, and you guys can read them as well as I can. There are 2 patents filed on the work that I'm going to tell you about today, so cross our fingers that those will be awarded and licensed, and we will make a lot of money, and we'll be able to retire in a Pacific tropical island where it will. Carol promised me that. Absolutely, yeah. Carol promised me when I was recruited to Minnesota that it was always warm and balmy, and there was tropical sea breezes, and so maybe when I retire. So, we live in an interesting and very exciting world, and there's an intense drive for personalized medicine in cancer. So, recognition that every tumor is different, every patient is different, and this has spurred the idea that every individual should be treated according to their own self and their disease. And so we now have developed these very sophisticated tumor classifications and have started pairing them with appropriate therapies. And those of you who were here for Doug's presentation at the beginning of the academic year probably remember the nature of the I-SPY trials and doing exactly that, giving the right therapy to the right patient for the right disease. And then pharmacogenomics. So every person responds to drugs differently, and Pam Jacobson here is a leader in this field. And so again, trying to select the drug not only to attack the disease, but to be compatible with that individual's genetics and metabolism. And then there's a newer area of personalized prevention. So we want to have actions paired with risk. And so, for example, some of the work that Steve is doing, trying to understand how to mitigate issues with people that are exposed to tobacco-derived toxins or toxicants. You want to make sure that if your actions carry any type of potential adverse effect, you don't want to expose people that are at low risk to those potential adverse effects. So we want to make sure that we define the right risk population to do our prevention actions. There's a lot of work going on in early detection, and early detection can then be paired with chemoprophylaxis, and then the burgeoning field of immunoprophylaxis. So essentially doing vaccines, much like we've used vaccines to prevent infectious diseases, to actually prevent cancer. And it's one of the really fun things about my job is that I get to be involved with all of these things, and it spreads me out way too thin, but at least I like what I do. All right, so what are the unmet needs in cancer testing? What are the things that are desperately needed? So we need accurate predictors of cancer risk. So we know that in humans, there's a lifetime risk of cancer of about 1 in 2 to 1 in 3, depending on whether you're a male or a female and who you believe. But it's pretty high, and yet 50% of the population or 65% of the population will not get cancer. And so it would be really good if we could focus our interventions and our emphasis on those people who will. And so how do we actually define risk? We also need robust tests for early cancer detection. And we have some really good methods out there, most of them based on imaging. There are some tests that have not stood the test of time. And I think that the experience with prostate cancer and breast cancer early detection illustrate both the power and the failures of these things. And so we really need better tests. It's not that the ones that are here are bad or terrible. We just need better ones. We also need biomarkers to prognosticate cancer progression. So sometimes you have cancer that is not particularly aggressive, and the treatment can actually be worse than the disease. And so as a man in his 50s, I know that I probably have prostate cancer. You know, if somebody goes looking for it, they'll find it. But the chances are that I will die with it rather than from it. And so if I don't need to be treated, if I don't need aggressive treatment that has a lot of morbidity, I'd rather not get it. And so we need to understand, you know, the word cancer is a loaded word. It carries a lot of social implications. And so we need to be able to remove the fear and the stigma of this word so that we can actually treat the disease accordingly. And then finally, as we develop all these really cool personalized therapies, we need to figure out who benefits from that. So I think a great example right now is all of this excitement in immunotherapy and checkpoint blockade, for example. And we know that somewhere between 20 and 50% of patients will respond to checkpoint blockade, which is great because otherwise they would die. But checkpoint blockade is not without pretty severe and toxic side effects. And so if you're not going to respond to checkpoint blockade, you're going to die from your disease and your quality of life is going to be miserable. And so most of the efforts trying to develop companion diagnostics so far have failed. It's not easy. It is definitely an unmet need. And so as we embark on research projects, we really do keep these things in mind. It's like, does anything we do address any of these needs and how? So I'm going to talk about dogs. And so for about 20,000 years, give or take, humans and dogs have shared their lives quite closely. And so we now believe that there were parallel domestication events. My feeling is that dogs domesticated humans repeatedly, but other people feel differently. And so there was something about these wolf or wolf-like creatures that made them very amenable to coexist with human communities. And eventually, they really became part of our lives. And in the last 50 years, they really become absolute family members. So the dogs have migrated into the house, they eat at our table, they sleep in our bed. And they serve a lot of purposes. And so in addition to our job as veterinarians to try to improve animal health and well-being, we can actually try to understand how they can help us improve human health and well-being. And so what are some of the benefits? I'm going to come over here now so that you guys don't have to crane your necks. It's their turn. So what are some of the benefits of studying cancer in dogs? And so one thing, as I mentioned, is dogs share our environment. They eat the same food. They have the same shelter. They swim in the same lakes. They walk along the same trails and so forth. And if you know college students who have dogs, they also tend to eat the same brownies and other, drink beer out of the same bowl, etc. So dogs get cancer. We don't have to give it to them. Mother Nature sort of does this for us. It's unfortunate, but it happens. And while the diversity of cancers that we see in humans occurs in dogs, the frequency of each cancer is different. And there are some cancers that are very intractable in humans because we don't see a lot of them. It's hard to study, but they happen very often in dogs. And so that's the case with osteosarcoma, which I'll be talking about today. Dogs are also organized. Over the last 400 or so years, people have bred them for form, function, or appearance. And that has created these really interesting genetic bottlenecks that have created different patterns of risk. And so studying how cancer happens in these different dog breeds can help us identify genetic risk factors that we have inadvertently selected for. And finally, the progression of cancer is adapted to lifespan. And I will be able to illustrate this really nicely. So that a dog lives, 10 or 12 years, and a human lives 70 or 80 years, or maybe longer. In the case of my parents, longer, thank goodness. And so when they develop cancer, the rate of progression, sort of the 5-10-year windows in humans, happen in 1 to 2 years in dogs. So we can actually get information a lot more quickly than we would if we were studying comparable disease in humans. So we can accelerate research by looking at dogs. particularly because of this speed. And so the generation time is shorter. We can get stuff completed much more quickly. There's large numbers of subjects. And because of the ethical approaches that we can use, because of the lack of accepted standards of care, because of a lot of societal norms, we can do ethical research in dogs to develop methods that will apply for personalized care. So dogs are large. Their cancers are treated with comparable types of medicines, comparable types of protocols. And if we have a new treatment that has passed the small animal test, we can actually bring it to dogs much more quickly and efficiently than we have to humans. And I'm not going to talk about that today, but we have done that repeatedly with a few products, both from the U of M and from outside the U of M. So I'm going to spend most of the rest of the talk talking about osteosarcoma, but it really illustrates the process. It's not the only thing we do. And so osteosarcoma happens in humans and in dogs, and the anatomic distribution is pretty similar. And this is a comparison and contrast. So in humans, osteosarcoma is a very rare disease. So there's less than 1,000 cases per year diagnosed in the US, and 75% of the diagnoses are in children and adolescents. In dogs, it's a very common disease. And so there's some very rough estimates, but some people believe that there's actually more than 80,000 cases of osteosarcoma per year diagnosed in the U.S. The published numbers vary from 8,000 to 100,000. Unlike kids, where this happens mostly at a young age, the disease happens in dogs at an older age. And so we do have to be very cognizant that the age distribution is different, and that means that when we think of osteosarcoma as a model, we have to be very careful about whether, in fact, the pathogenesis, the risk, the progression is similar in both species. In humans, many cases of osteosarcoma are syndromic, so they occur in kids that are predisposed because they have heritable risk factor like Li-Fraumeni or one of several others. For the ones that get the disease sporadically, the risk factors are uncertain. In dogs, there's very well characterized breed predilections that are partly associated with size, both body mass and height. But there's certainly a lack of characterization of what those factors are, whether they're only the drivers or size or whether there's other things. And when you look at the distribution of the data, it's probably safe to assume that the drivers of dog size are some of the factors. It's not as simple as IGF-1 that's been tested repeatedly. But there's other things because there are breeds that fall off that risk to size or risk to growth rate curve. In both species, the disease causes high morbidity and mortality, and death is almost always due to metastatic spread of the cancer. So in dogs, the metastatic pattern looks very much like it would in humans. This is a radiograph of a dog with a tumor in its distal front limb. And the tumor basically goes from the front limb eventually to the lungs. And so this is a radiograph of the lungs, and you can see these shadows that represent lesions. And this is the same dog after it went to necropsy, and you can see these large tumors that have taken up the lung surface and basically just consolidated and solidified it as bone. So you can imagine that this is not a very good breathing surface. It's also excruciatingly painful, so quality of life is a problem. So I'm going to spend some time telling you how we developed this prognostic test for osteosarcoma and really illustrate how dogs can be contributors or participate in the development of personalized medicine. So the first paper that we published in this aspect was published in 2011. This is work that actually was started by Catherine Gavin, who was a very talented undergrad at Augsburg who was accepted into medical school and took a year off to come work in the lab doing bone cancer. And it's really gratifying because Catherine became so excited about the work that she was doing that she's now a pediatric orthopedic surgeon treating kids with osteosarcoma. And at the end of the first year, it was taken over by Malka, and so Aaron worked very closely with both Catherine and then with Malka, and really this project was born out of that collaboration. So for this project, what we did is we had some funding, and I won't get into the details for how we got that funding, but we had funding. And we had a large archival collection of samples from dogs with osteosarcoma, and we had tumors, whole tumors, and we had cell lines. And Catherine and Aaron and I spent a lot of time trying to decide whether we should do gene expression profiling on the tumors or the cell lines. And ultimately, we chose the cell lines. And I suspect that we probably chose them for all the wrong reasons, but it ended up being the right choice. It ended up being the right choice because the cell lines did two things for us. One is they originated from dogs, and they removed all of the stromal elements from the tumors, right? So we have these pure populations of tumor cells. And so the noise when we actually looked at what was happening intrinsically in those cell lines was very, very low. And we were able to do gene expression profiling on an ancient array platform. So this is really legacy data. And allow the data to tell us what was going on instead of trying to force the results into some preconceived notion of what should happen, which really is, I think, the death of these large bioinformatic approaches. And so I'm going to be showing a lot of heat maps today. And for those of you who are not familiar with heat maps, for virtually all of them on the top axis, so basically the columns are going to represent individual samples or individual patients. And on the horizontally, so the rows are going to represent expression of individual genes. And so what happened is when, after we did all the QC, what we found is that if we looked at unsupervised clustering, we just let the data sort themselves out according to similarity. We found that the samples sorted themselves out into two very distinct groups where the differences were driven by two gene clusters where the genes moved very, very closely together. So there was this gene cluster here and this gene cluster here. And obviously you can tell that hopefully there's no one here who is red-green colorblind. So this is red, this is red, this is green, this is green. And this group is very, very different from this group. And we started trying to figure out what was different between these groups. One of the first things we noticed is that we had one normal sample, so one sample of normal bone cells that we had collected serendipitously. And it basically anchored this group here. So all of these tumors in this group looked very much like normal. And all of these tumors here looked very much like not normal. And when we went through and looked at all of the possible variations or variables that we could analyze, So when we actually looked at the genes, this group here represented a cluster of cell cycle genes, mostly in the G2M transition or DNA damage repair. And this cluster of genes is something that was kind of ill-defined, and so we just baptized them as microenvironment genes. They tend to do things that allow the cells to interact with their surrounding environment. And when we looked at what was different between the two groups, what we found was this significant association with outcome. And this association was independent of treatment. So the dogs, obviously the dogs that get treated do better than the ones that don't, but if you separate the ones that get the same type of treatment, this relationship always holds. And so this is for dogs that were treated with a standard of care of removal of the primary tumor by limb amputation followed by chemotherapy. And this group here, that we call molecular phenotype 1, had a relatively long survival, as you see here. And this group here had a pretty dismal survival. And so this told us that, in fact, there seemed to be a difference between these groups in terms of what would happen at the level of the patient. So fast forward a number of years, and we started a new collaboration that now included David Largaspada, Logan Spector, Subri Subramanian, Brenda Weigel, Brandon Moriarty, and a number of other people, and a really strong bioinformatics team. And so in this case, we were able to actually expand the comparative study to humans and dogs. And so originally, we actually were able to document in our previous study that the molecular signature we discovered in dog cell lines was present in dog tumors. So if we knew what to look for, we could find it. And it was also present in human tumors, and the prognostic significance was retained. So we were then able to characterize the osteosarcoma signatures in greater depth as part of the Soviet osteosarcoma project. So this is a really nice injection of both people and financial resources that allowed us to do a lot more work. And the outcome of this was that we identified several new conserved gene clusters, much like the clusters I showed you before, including 2 highly significant immune clusters. And this could be the subject of 17 lectures. So I'm going to really abbreviate it for the sake of time and to tell you how this is getting us to the next place. So these are data from a paper that is under review. And again, the leads in the group are Maka, who's the first author, and Aaron, who is the corresponding senior author, and then a lot of people in between. And what we have done here is we have taken samples from humans, mice, and dogs with osteosarcoma, each one also including cell lines that anchor the analysis. And these also are heat maps. And what you will see here is that the color scheme is a little different. So yellow means low, and yellow means high and blue means low. And there's a blue arrow showing where the cell lines are. And so the first thing that I want to attract your attention to is that the cell lines have areas that are deep blue. So there's genes over here in the cell lines that are not expressed. And we interpret that to mean that those genes are almost certainly associated with stromal elements, things in the tumor that are not tumor cells. And when you align these samples, again, in an unsupervised approach, you can see that there are these clusters that move very, very tightly together. And what we have done here is we've color-coded them because it turns out that this cluster of genes that's labeled in green here is the same as this cluster of genes that's labeled in green here and this cluster of genes that's labeled in green here and so on. And so what we found is that there are, in particular, a cell cycle cluster that's essentially identical to the one we found before. And it's called 4 in human, 3 in dog, and 8 in mouse. And so these are color-coded. And then these 2 immune clusters that actually make are 2 parts of a very large conserved cluster. But each one is driven by slightly different cells. When we look at the cells that make up the immune cluster, and I know Aaron's going to frown because he hates this slide, but I love it. I just think it's very, very cool. What we've done here is we've separated human, dog, and mouse. And using a signature that allows you to define or at least predict the cell type that's present based on the transcripts, we can actually see that there are certain samples, okay, so in this case cell lines, for example, where you see absolutely no immune signature. And there's some samples where you see a lot of immune signature, and that's true for dog, human, and mouse. And when we see a signature, the main drivers are monocytes, macrophages, and T cells, but every other cell type is represented. And one of the things that's really important is that no single one cell type seems to be the driver of the effect. So all of them tend to move together. And this is very counterintuitive because for the most part, there's been this concept that separates macrophages as good for the tumor and T cells as bad for the tumor or good for the patient. And what we have found initially in osteosarcoma is that these 2 cell types actually move together. It didn't separate out into more macrophages good for the tumor and more T cells bad for the tumor, which is a very interesting idea. Okay, and you can actually take all of those genes in each cluster, and you can create a summary score of all the values of the genes. And I won't go into detail on the methods, and Aaron and Malka are here and they can explain it. But basically what we've done is each dot here represents a single individual. And the gene cluster expression summary score is the combination of all the values in the genes in that particular cluster. Okay? And so for any cluster, we can define, in this case, these are illustrated by immune cell clusters. So cells basically have very low immune clusters. Transcripts that represent immune cells are not present in osteosarcoma cells. Okay? Biopsies can actually be divided into low immune scores or high immune scores in each case. And you can see that the division between low and high is essentially whether they're a negative value or a positive value. So we don't have to assign some arbitrary number of, 10 is low and 1,000 is high. Anything below 0 is low. Anything above 0 is high. And when we actually look at what happens when we divide the samples according to those values, again, we have our clusters, right? So immune cluster 1, immune cluster 2, cell cycle cluster for humans and for dogs. And the way that Aaron described this is there are some clusters where a lower summary score means poor outcome, and these are the immune ones. And there are clusters where the high summary score means poor outcome, and this is the cell cycle. And if you divide the samples or the patients based on quartiles of the distribution of the summary score, you can draw some comparisons. And so, for example, here, low immune infiltrates in these tumors would predict poor outcome, and that's statistically significant. And in this case, high expression or the highest expression of cell cycle would predict poor outcome, and that is significant. And so that's illustrated in Kaplan-Meier curves shown here. And the one that I really want to draw your attention to is this one, because I think this is the easiest one to see. Okay? And so this guy represents this guy right here. So immune cells, low immune cells, meaning a worst outcome. And what you can see is that in this group, okay, when we compare the first 3 quartiles of infiltration versus the lowest one, the lowest guys die. This is a really bad outcome. But the higher ones do great. These guys don't die. Okay? And you don't need statistics to see that this is different from this. And so it becomes a really powerful tool to be able to predict how a patient is going to do based on what genes are expressed in the tumor, and in this particular case, telling us about the immune microenvironment. So I want to illustrate another thing in this particular slide, and that's that this top graph is looking at human cases. And you can see that the y-axis in this Kaplan-Meier, which is represented in days, goes out to 3,500 days, or about 10 years. Okay? And the lower one is dogs. So we're looking at, I think, this one right here. Okay? And this goes to 300 days. So 10 years versus 1 year to essentially get the same data. So again, it just illustrates the power of being able to do stuff much more quickly. So what have we learned so far? We have learned that we can classify osteosarcomas, and I don't want to steal the thunder from Aaron and the group, but this actually applies to a lot of other tumors. But I'm going to focus on osteosarcomas. We can classify them into groups that have significantly different biological behavior. The behavior of the tumor cells and the associated stroma, at least transcriptionally, is conserved across species and strongly correlated with the natural history of the disease, meaning progression, metastasis, and overall survival. And the testing could be used to personalize therapies. I'm going to move a little faster because we started late and I don't have a lot of time. So why are these tests not being widely adopted? Well, unlike therapies, diagnostics require huge numbers, or therapies are not easy to approve. Diagnostic tests are actually harder. So we need to go through the process of FDA approval, and this needs to be validated in large populations. And in osteosarcoma in particular, there is no such thing. There are no large populations of patients in which we can validate this. So that's going to be a perpetual obstacle in humans. It's a different thing in dogs. And so we need to find a partner to license that, and we're working on that with a company that actually does diagnostic testing in dogs. Another thing that we take blame and responsibility is that we have not been aggressive in disseminating this information. So we've published papers, and the people who read the literature know about it. But we haven't actually taken out to the world and blared out in a bullhorn that we've discovered this. And I guarantee you, if we were at a different institution, this would have been done, and this test would be much more widely talked about. So we have to be better about tooting our own horn if we believe that the data are strong enough. And finally, there's another obstacle, which is we've relied on gene expression platforms. And these gene expression platforms, especially RNA sequencing, are definitely feasible for individual patients, but they're not yet routine. And so we need a more user-friendly platform so that this can be implemented in practice. So what are potential solutions? One is we could actually take the gene clusters that we have and identify a core set of genes that when we measure them, whether it's 5, 10, 15, 96, they give us the same type of data that we see with RNA-seq. Another potential solution is to expand or adapt this method to look at biomarkers in blood, again, trying to overcome the issue with biopsies. So how do we select relevant and user-friendly cancer biomarkers? And so we need to understand whether the molecular phenotypes that we find are stable over time and in vivo. And then can we identify biomarkers that detect disease? So in an animal or a person that doesn't have clinical signs, can we find if the disease is present? Once the disease is there, can we identify biomarkers that prognosticate the disease? And can we then identify biomarkers to measure response to therapy, to personalize these? And so we've started tackling these issues in 2 papers that we published recently, end of 2015 and middle of 2016 or end of 2016. And again, it's the same team that you can see. And so the first thing that I want to show you is that when we look at our cell lines, at least their molecular signatures are very, very stable over time. And so these data are from the original paper that I showed you. These are experiments that were done in 2009. These are experiments that are done about 2013 or 2014, using essentially the same material after many, many passages. And so this is part of the cell cycle signature. Molecular phenotype 1 is low, molecular phenotype 2 is high, so highly expressed cell cycle genes, a much lower expression here. And when we do the experiment again in molecular phenotype 2, you can see different array platforms. So we're actually using a more advanced array platform, but still legacy now. The genes are highly expressed in molecular phenotype 2. And the cool thing is if we actually treat these cells with DNA and chromatin modifying drugs, things that change the transcriptional landscape, we can make these molecular phenotype 2 cells start behaving more like molecular phenotype 1 cells. So not only are the gene expression patterns stable, we can quite modify them. And very predictably, if we do chromatin immunoprecipitation, we can see that the effect or the predicted effect of chromatin modification affects the molecular phenotype 2 much more strongly than it does molecular phenotype 1. So these things are stable and they're not fixed. We can change them. We can put our cells into animals and they behave in a very predictable manner. So this is our original hierarchical cluster. This is unsupervised. So in this case, we've sort of flipped it around. Molecular phenotype 1 is on your left and phenotype 2 is on your right. And we've selected cells from this molecular phenotype. These are the good guys, the less bad guys, I guess. They're very, very challenging to grow, so we don't have a lot of representatives. These guys grow like weeds, so we have a lot more of these. We can put these into mice in the orthotopic setting, so we can inject them into the tibia of mice, and most of this work was done by Hiro. And all of them grow, but you can see from the luminescence signals that these guys here grow incredibly fast. These guys grow faster. And these guys grow much more slowly. And the reason that you have an X here is because we had to sacrifice these mice because of their tumor burden. So molecular phenotype 2 behaves more aggressively, molecular phenotype 1 less aggressively. And interestingly, there was this really cool thing that we found that within a few hours after we injected the cells, when we imaged the mice, molecular phenotype 1 basically just had tumor in its leg. But these mice that received OS2 or OSCA40, all of them in multiple different experiments had the lungs lighting up. So this was really, really curious. And even more curious was the fact that by the end of the first week, we could no longer detect the cells. So these cells had either showered the lungs and then left, or they had showered the cells and a few of them had remained, or many of them had died. So we don't know if this is below the level of detection, or if this is because they go to the lungs and then they leave. The lungs are not prepared for them to yet do metastasis. But we can actually infer that the tumor cells leave the intraosseous environment because we're injecting them right into the bone cavity, and they home to the lungs because we don't see them going anywhere else. They're not accumulating in the liver and lymph nodes and the brain. They're just going straight to the lungs. So there's something peculiar about these cells and what they like for the environment. And it turns out that as you follow the animals, if you look, the cells from these more aggressive phenotypes have a greater propensity to metastasize, and they do so earlier. So we know that these guys can metastasize, and these guys also can metastasize, but if you look collectively, these guys metastasize more and sooner. Okay? So this is what the tumors look like in the animals, and they're very, very hard to tell apart. So in the terminal stage, grossly, the tumors are not that different. Microscopically, they are quite different. So these tumors look more bland. There's more organization of the osteoid seams that they're laying out. There's fewer proliferation. These guys are just going crazy. They're invading. They're doing a lot of bad things. And when we look at the lungs in this case, again, molecular phenotype 1 is much less metastatic. So in this particular case, we do not see any evidence of metastasis. Molecular phenotype 2, we see a fair number of microscopic metastases that you can see right there. This area of tumor growth in the lungs. And the tumor basically looks similar to what we saw in the primary tumor. So how can we use this to our advantage? So the innovation is that we now have the xenograft model. We can take the tumor and isolate RNA, and we can do this really cool thing, which is because the tumor cells come from the dog, and the environment comes from the mouse. We can actually develop a method to test which is which. Okay? And so what Malka and John Garvey did is they constructed a hybrid genome that actually includes sort of a stapling of the dog genome and the mouse genome. And using some very nifty bioinformatic tools, they were able to identify sequences that originate from dog and those that originate from mouse. And so if we look at all of the sequences of a tumor mapped to both, we can see that the things that are most different, okay, or the things that tell us what drives, if we compare cell lines to tumors, is that all of the tumors have a lot of mouse transcripts in them, as you would predict. And so if you look at all of the genes together, the things that make cell lines and tumors most different is this mouse contribution. Okay? And what you get is tumors derived from OS1, tumors derived from OS2. They separate nicely, but these are more similar to each other than the cell lines, which only have dog genes. So all of these things that are mouse are turned off in the dog. Okay? Conversely, if we only take the dog genes and we see what happens using the same approach, it becomes really interesting. Now, in this case, each cell line pairs with its tumor. Okay? So this is OS1, cell line tumor. This is OS2, cell line tumor. And remember, we're only looking at the dog gene, so we've filtered out all of the mouse stuff. And it tells us that the cell lines, okay, the gene expression profiles of the cell lines, are actually quite stable in vivo. So they're retaining the same signature that they had originally. When we actually look at the signal of only the mouse genes, and we see what the microenvironment is doing. So we're looking at clustering with statistically significant mouse genes for both xenograft groups, and then we're basically taking those gene clusters and looking at what pathways or functions are activated. And so the first thing that you see is that, as I showed you before, the mouse infiltrates into these tumors create two very, very distinct groups divided by OS1 and OS2. So these are mouse replicates. And so what we find here is very consistent. And what we find enriched in OS2, okay, so overexpressed in OS2, are things that deal with how the tumor interacts with cells of the immune system, even though these are nude mice and they don't have T cells, but they have the rest of the immune cell. And so there are a lot of sequences in the mouse that basically are telling us about organization of the tumor and its interaction with leukocytes. What's enriched in OS1 or depleted in OS2 are things about essentially mesenchymal cell development. So things about metabolism and organization of muscle, bone, etc. So very, very different types of behavior at the gene expression level from the mouse stroma. Okay? So what's missing? So, as I said, these are nude mice, so T cells are missing. And it's just interesting that the stromal cells in OS2 tell us that there's a favorable environment for Th17 cell differentiation. So it seems like the stromal cells here are screaming for Th17s to come into the system, but they're not there. And so we think that what happens is the signal gets amplified. Okay, the cells scream, Th17 come, they don't come, they scream louder and louder. And so we actually see this amplification. And we recognize that if T cells were there, this would alter the landscape. And it's something we have to keep in mind. Okay? So what have we learned? We have these osteosarcoma cell lines that we can use as model. They're derived from primary explants, and they have stable molecular phenotypes. The xenografts recapitulate the behavior of the original tumors. And the xenograph models with really robust bioinformatic methods allow for efficient separation of tumor-derived sequences and host stromal sequences. And the tumors, quite predictably, show diverse interactions with the stromal environment that are probably associated with behavior and outcome. Okay, so where do we go from here? Does the information address at least one unmet need? Yes, it addresses an unmet need of prediction and prognosis. So how do we apply this new knowledge to overcome obstacles and deploy our tests? So now we can use this information to develop a user-friendly blood-based platform for biomarker discovery. Okay, so bear with me. We've got about 5 minutes. Okay, so how do we apply this new knowledge to our advantage? So we now want to test this hypothesis. We found that there's these distinct tumor-stromal interactions. And we believe that these interactions are actually going to be reflected systemically. So we can go outside the tumor and still see differences. And we want to test the hypothesis using a blood-based diagnostic approach. Okay, so how can we use blood-based diagnostics for tumors besides traditional biochemical markers? So 2 platforms that are in vogue right now are circulating tumor cells. So this is an FDA-approved platform for certain tumors. And it actually is quite useful. In the case of osteosarcoma, there is one paper that was published out of Mayo Clinic more than a decade ago about looking for markers in circulating osteosarcoma cells. So we know that the cells must be there because they go from point A to point B. But the data from Mayo have not really been reproducible. So whatever they did to try to find it, I think, was a flaw in flow cytometry. The other platform that is actually quite complementary is to look at exosomes and microvesicles. And I'll tell you more about what these are in a minute. So this really nicely illustrates the 2 things together and how they're complementary. And so this is from a very recent review in Cancer Cell. And what they've done here is they've illustrated pancreatic cancer and melanoma. Both of them are vascularized. And into this vasculature are released circulating tumor cells that are depicted by these guys here. And exosomes or microvesicles that are depicted by these guys here. And the exosomes actually increase vascular leakiness, and they interact with cells in the target organs, be it sentinel lymph node. Can't really see it very well here, but they're supposed to be a lung here, and they're supposed to be a liver here. So in the liver and the lung, they interact with the resident cells, and they create a pre-metastatic niche, a niche that the circulating tumor cells can colonize. And ultimately, they alter and improve the metastatic niche, so they improve vascularization, they keep immune cells at bay, et cetera. And so these 2 guys work in concert to actually create the metastatic process. Okay. So what are exosomes and microvesicles? They're ubiquitous, small secreted membrane-bound vesicles that originate from the endosome and the plasma membrane. Okay, so there's very different kinds. Some of them come from the endosomal pathways, and some of them don't. These vesicles carry membrane and cytosolic proteins, lipids, and RNA that mediate intercellular communication. So it's a way, it's kind of like a telegraph. Cells from one place communicate with cells at another place by secreting these things and sending the information they want in these small packets. And tumor cells and immune cells are very prolific producers of exosomes. They make a lot of exosomes. So what we've come up with is an innovative method to identify tumor-associated exosomes. And so when we take an animal that has a tumor, the tumor-derived exosomes are mixed with other host-derived exosomes, right? So all cells in the animal or in the person are making exosomes. And it's really hard to figure out which is which. So you can do biomarker discovery by using brute force. So you take a large number, very large number of patients and a very large number of healthy individuals, and you look at everything that's there and you try to sort out what's in the patients and not in the unaffected individuals. But you still can't figure out where the stuff comes from. Okay? And you can do that using a variety of omics methods. But we think a more clever approach is to use the species mismatched samples to identify the origin. So what we've done here is first, can we identify osteosarcoma exosomes? And so this is work done in Brad Bryant's lab. Brad's a collaborator down in Texas. And so he's isolated exosomes from osteosarcoma cells, and these are electron micrographs showing basically these little round guys that conform to the size and shape of exosomes. And they express these tetraspanins that are characteristic markers of the endosomal pathway-derived vesicles. Okay, so we can get things that look like exosomes. So what is in those exosomes? So we have osteosarcoma cells. We enrich the exosomes. We isolate the RNA. And we essentially do the same type of bioinformatics that I showed you before. Right. I'm going to show you tumor in a minute. These guys are from cell lines. Okay. And so what we find is that using the same approach that I showed you before, this is looking at a fold change between cells, cultured cells, and exosomes. Okay. So how is it different? What's enriched or depleted in the exosome as compared to the parent cells? And what we find is that there are three clusters that are different between OS1 and OS2 cell lines. Okay? Cluster 1, when we look at all the genes, they don't really fit into any known pathway. And then cluster 3, which is enriched in OS1 as well, is essentially metabolic and cellular signaling. So a lot of things having to do with energy-producing compounds. Okay. When we look at this middle cluster, which is enriched in OS2, we see two things. We actually see some metabolic signatures as well, as illustrated here by LXR-RXR, but we also see transcripts that are involved in communication between innate and adaptive immune cells. And so, again, it's telling us that not only in the local environment of the tumor, but also distantly, these OS2 cells are being very, very aggressive or very active in modulating the immune response. Okay? So do these exosomes do anything to cells that would create the niche? So what we've done here is we've taken exosomes from our cell lines and added them to other cultured cell lines, either fibroblasts or endothelial cells, and measured what happens. And so these are the original primary cells. What we've done here is we've taken CD63, which is a tetraspanin, and linked it to GFP and transfected the cell stably to express CD63 GFP. And so you can see the endosomal pathways lighting up, and these guys get secreted. So we actually collect the exosomes, and we add them to cells, and this is what the cells look like in bright field at time 0, and after 24 hours, and you can see that there's no GFP at time 0, but after 24 hours, we see nice incorporation of the exosomes that are labeled with GFP into the cells. Okay? And so again, Brad's group looked at what happens in terms of function and whether they look at endothelial cells or fibroblasts and whether they look at migration of those cells on a Boyden chamber assay or a standard two-dimensional scratch wound assay or cell numbers. You can see that adding the exosomes increases migration and increases cell numbers, and that O40 here, which represents OS2, always has a greater magnitude of effect than O32, which represents OS1. So the exosomes are changing the behavior of these fibroblasts and endothelial cells in culture. Okay? And so are the gene expression patterns of those cells changing as well? So the experiment here is basically the same. We take osteosarcoma exosomes. We add them to mouse lung fibroblasts. We separate RNA, and we do bioinformatics using those RNA species. And when we look at fibroblasts that are exposed to OS1 exosomes, there's a number of genes that are significantly different. But these guys don't sort into any pathway that we can tell. So there seems to be just a random assortment of genes that are there, and we don't really know what that means, if anything. These might all be essentially random findings. When we look at OS2, there are very clear pathways that are enriched in the genes in the fibroblasts. And interestingly enough, the thing that starts coming up again is what the fibroblasts are doing to Th17 responses and other immune or innate immune pathways. And one of the very interesting things is that unlike in the exosomes themselves, where we saw a collection of genes that would favor Th17 responses, in the fibroblasts, we actually see the opposite. And we have no idea what this means. This is going to require a lot of digging to try to sort it out. And so what's our process for discovery? So we basically now know that we can establish xenografts. We can collect serum from those animals, so from the mice. We can purify exosomes, and we can isolate RNA. We can do RNA sequencing, and we can align the sequences to our hybrid genome to calculate gene abundance, and basically establish and validate donor and host biomarkers, things that are enriched in the serum of the animals with the xenografts that are attributable to either the tumor or the host. And I have to thank Sarah Myron for essentially the concept of this slide. She's our patent attorney, and this is one of the things that she came up with. And then Malka helped me yesterday to make it much more user-friendly than what Sarah had done. And so if we look, now what we're looking at is a heat map of serum-derived exosomes from the mice that either have xenografts or that were injected with PBS into the bone. Okay, so sham versus xenografts. And so the controls are sham, and these are exosomes from mice that have growing OS1 or OS2 xenografts. And when we do the analysis, you can actually see that there are 7 different clusters of genes that we can define based on this heat map. And the lowest cluster here is all mouse genes. All of these guys are dog genes. Okay? And these are the correlations for the genes that comprise each cluster. And so, so there's at least 4 of them that are very, very robust. 2 of them that are weaker, and the mouse genes don't really have a lot of correlation, as you can see here by the smattering of colors. And we're especially interested in cluster 1 and cluster 4. So cluster 1 can help us identify the presence of tumors. So it's always high in the animals that have tumor, and obviously never present in the animals that don't. And then cluster 4 can tell us something about biological behavior, meaning that OS1 and OS2 are quite different. And so what's present in those clusters? What are the pathways that are enriched by those genes? And so in cluster 1, we have actin cytoskeleton, protein kinase, a few other things. And then in cluster 2, again, we have some really interesting things about the exosomes trying to modulate what's going on with the immune system. So ICOS and ICOS ligand signaling, PI3 kinase, et cetera. And what are the drivers of those events? So not surprisingly, for things that really distinguish tumor from not tumor, we tend to see events that are driven by P53, among other things. And by things that distinguish OS1 from OS2 in the exosomes in the animals. Remember, this is the xenografts. These are the serum from the mice, okay, that have tumor 1 versus tumor 2. We see signatures that include interesting genes. And when you actually look at what these things do, they seem to be very involved in stress-resistant, the unfolded protein response, and adipogenesis, things that we already know are involved in tumor progression. So this is as far as we've come. We have gobs and gobs of data that are still waiting for more analysis. So what are some of the next steps? So one step that's essential is we have to actually take these biomarkers that we've discovered in mice and validate them in the target species. So we have to go to dogs with and without tumor. We have to collect exosomes from their sera. And we have to actually show that the mice inform what's going on. So in those exosomes, we should be able to isolate signatures comparable to the ones that we've had, and we should be able to tell the dogs that have tumor from the ones that don't. Moreover, we should be able to tell the dogs whose tumors are going to be really, really awful from the ones that are not going to be so bad. And presumably, we can actually correlate that with what happens in the tumor biopsies themselves. We want to extend this and validate this xenograft models for early detection, for example, in human colon cancer. So we want to create essentially the same type of model for colon cancer that we could actually use to pair up with patients that go to get colonoscopies. So at the time you get a colonoscopy, you can get a blood sample, and you can get a prediction of whether those polyps represent something that's likely to form tumor, not likely to form tumor. Might there already be tumor there, and what's it going to behave like? And finally, we need to be able to modify our model to allow for syngeneic systems, right? Because T cells aren't there in these models. And T cells are a huge component of how the tumors behave. And so some of the work that we're doing right now, and Alicia's not here, she might be over at MCRB, but Alicia's a postdoc in the lab that's working on a project that really feeds into this, is to actually modify the system so that we can use physical means to separate the exosomes and be able to tell what's going on in a system that's immunoreplete. And finally, we believe that there's a lot of people who actually would want to use the system to test, like, is my drug working? Is my drug not working? You know, how can I detect if a smoker is likely to get cancer or a smoker is not likely to get cancer? So there's a lot of really cool things that we can do with this model. So in summary, what I hope to have conveyed to you is that we identified a very cool, very robust, prognostically significant gene expression signature for osteosarcoma. This actually extends to other tumors. Translation of the signatures is not impossible, but it faces some challenges. So we're not giving up. We believe that we can actually make this into a test, but it's not as easy as just handing it over to somebody else. We've expanded the foundational methods that we use to do this research to develop a platform for discovery of blood-based biomarkers. And this platform has multiple applications, but there are some limitations that we have to be aware of because the hosts that we're using are immunodeficient, and so we do need to expand this to syngeneic models. So I want to take just a minute to recognize the people who did the work. This is a picture of my lab, obviously not in the winter. Malka, who I mentioned repeatedly, and Hiro, who did most of the xenograft work. Obviously, all of the other people in the lab who've done a lot. So Patrice, John, Ashley, and Katie, who've really supported the work. More recent picture, now it includes Malka. Kelly, who's working on the immune components of osteosarcoma, so she's leading really cool work on a clinical trial. Alicia, who I mentioned, who's working on the exosomes. And again, old hats that seem to never leave, Patrice and Katie and Ashley and John. They seem to have been here forever. Some new additions. Abby, who's an undergrad in the lab. Taylor, who's working on early detection platform. And Goldie, who's making his first appearance with our lab. It's really cool. I don't have to pay him, so or her. I don't know. And this is Brad's lab. So Brad, Brian, Clarissa, who did a lot of the work that I showed you, and the osteosarcoma group, and our collaborator Subri, who's not pictured, Logan, David, Malka, and Aaron, who really has been a stalwart for this project and has been a huge driver for everything we do. And there's many, many more collaborators that I can't show you. So people who've paid the bills. And that's my little girl, and happy to take any questions from you. And apologies for going long. We'll blame it on the snow.
Cell cycle and immune gene sets, and the idea that it goes to the bone and then AKT comes on. I mean, to me, it screams hormone action. And so I know it's hard to see that in most American animals that have been spayed or neutered, or in kids in pre-puberty. Are there any correlation with sex or sex hormones with osteosarcoma, or genes in your gene set that are steroid receptors?
So the question is whether there's any correlation with sex or sex hormones. So there's definitely a male predilection in kids, and it's not necessarily repeatable, but people have reported the same predilection in dogs. I think it's more 50-50.
Did I see ER alpha in your gene set in the last slide?
I don't recall ER.
SR1.
Yeah, but anyway, the short answer to your question is we haven't looked. And I think that it screams potential collaboration. So, Aaron, do you have a comment?
No, I was just going to say that I think it was estrogen receptor was a predicted activated target.
That's what I would have predicted based on what you're...
Yeah.
Sure. Yeah. Cool.
So I think we haven't looked, and it's wide open.
Other questions?
I thought the comparative approach was very novel. Have you merged human and mouse gene sets yet to make a hybrid genome?
So the question is, the comparative approach is cool, and have we done this in humans? And so Subri actually has some data. We haven't analyzed it using this specific pipeline that Malka and John Garvey developed, but it is available for us to do that. And so the idea of taking xenografts and trying to separate the stromal components and the host or the tumor components is something that a lot of people are doing. And so what we submitted for intellectual property protection is really our approach. It's not the concept, because the concept is not necessarily novel. The really cool thing, I think, is that we were able to expand this not only from the tumor, but really into the exosome compartment. And there are some very, very tricky things about doing that, and there's going to be some technical challenges to be able to do that effectively. Again, we have not done the human on mouse, but there's no reason to believe that it wouldn't work. In fact, it should work even better because the annotation on the human genome is so much more complete.
Other questions? All right. Thank you, Hyman.
Thanks.
Great talk.